Okay, thanks, that's fine. We had some delay, but it was not me. So we are starting, and I'm delighted to tell you something about the application for PCR in pork detection, mainly for halal issue. And as you can see in my working title, I will mainly speak about pork, but everything is valid as well for other species like horse, camel, poultry, and so on. So where do I have to show this in front of it?
No, it's
working. Okay, so when talking about PCR a few years ago, or even a decade ago, it was very difficult to explain people in the food laboratories why using this complicated technology and what PCR is. So today, as you can see here from the sign of the test center, or even outside, there was just a service laboratory. Everybody worldwide has used the word PCR, and even children knows that worldwide that PCR is somehow related to some unpleasant swabbing in the nose or in the mouth. And grown-ups will know PCR is a useful tool just to detect the COVID-19 or SARS-CoV-2 virus, to get a certificate, having some CT values, that this technology takes approximately 2 to 3 hours, depending on the samples, is a little bit more expensive than lateral flow, but is very specific and the gold standard for virus detection, detecting, for example, SARS-CoV-2 and not influenza. So PCR stands for polymerase chain reaction, meaning specific amplification of DNA, or for viruses, RNA, for example. And the technology used being today mostly is real-time PCR, and the abbreviation is qPCR. The difference is that there, in addition to specific primers, also fluorescent probes are labeled and being detected in this immediately in real time. So, sorry, I have just another small issue. Okay. So on the right side, you can see this iconic picture of a virus, but one can use PCR not only for virus detection, but also in food and feed analysis. As you can see in the middle for plants, so detecting plants, or the left cartoon for animal species or bacteria. Just for the abbreviations, RT-PCR stands for reverse, not for real time. So if this stands on your certificate correctly, it should be RT-qPCR for quantitative PCR. The cells in the plant cells and the animal cells, they contain the eukaryotic cells contains a nucleus, which means there is one genomic set of genomic DNA per cell. So it is defined, the amount. And one can use, for example, a single copy nucleus DNA for quantification. Therefore, it's also mentioned quantitative PCR. Or the cells has also multi-copy mitochondrial DNA, which is located in the mitochondria or in the plants in chloroplasts as well. So, very important for the detection of animal species, there are two possibilities. The nucleus single copy genomic DNA can be used for quantification. The mitochondrial multi-copy DNA for more sensitive qualitative detection, because a huge amount of highly conserved mitochondrial DNA is in the animal cells, but it might not be used for quantification because the number is not really defined. And usually one is using some sequences like from cytochrome B or cyclooxygenase as targets for animal species detection, because the species, that's really what PCR can detect really very specifically. So PCR is a suitable detection method for the species, which can be the pathogens like Salmonella, E. coli, viruses, but also the plants in allergens. We talked about plant allergens yesterday, the animal species or genetically modified organisms. And that's just a picture of a typical food. So the question could be, is there pork, this prova, in the meat And you can see here in the salamis, there's already a challenge can be seen. There's fat inside. And the question could be, are there some hidden processed compositions of, for example, pork meat in this food? So for all kind of PCR, one have to do a sampling. For example, for COVID, you know, the swabbing in the nose or in the mouth, a sample preparation, RNA or DNA preparation, a PCR setup, and finally the data interpretation. So the sampling is really the crucial step for all kind of technology, whether you're talking about LC-MS or ELISA or lateral flow or PCR. And for food, one is using a small amount, let's say kilograms, up to grams from the sampling, will have finally approximately 50 to 100 microliter of the DNA, and will use 5 microliter of the DNA for PCR, which means the test is only as good as it is representative for the sampling. So the sample must be homogeneous and representative. One should not forget before discussing the final results. That's really sampling is crucial. And for PCR, after using these representative sampling, one starts usually with 50 to 100 milligram for sample preparation. One can do the DNA preparation, which means lysis of the cells, having access to the DNA, to purified DNA without inhibitors. One can do this manually. You can see the left side, the second picture on top, or with automation. Then one is doing a qPCR setup in a dedicated laboratory, which has to be clean in terms of molecular biology, not of microbiology, and will run a PCR run, which can be done on a lot of thermocyclers. So this will be just this last slide to explain the PCR. In this qPCR, you will see on the left side or on top, the so-called thermoprofile with 2 temperatures, 95 degrees for denaturation, melting of the DNA, and for example, 60 degrees for annealing elongation. And the yellow dots are the measuring. And in each of these cycles, the DNA will be amplified or duplicated. So if one starts with one single DNA copy, one will have 2, 4, 8, 16, 32, and so on. And with 30, 35, or 45 cycles, one have millions and millions of DNA, which are labeled in real-time PCR. Just looking anti-clockwise is in such a thermocycler will show the results on the top right side. So that's a picture which you should remember as a minimum take-home message. You can see these blue dots. This is, let's say, the background measurements, the signals of the background. And this is more or less looking like a line, but it is not a straight line. The black dots are positive DNAs, let's say, or they are positive results. And the computer will differentiate this with a so-called threshold and will calculate with an algorithm a curve. So usually you will see these kind of curves. On the left side, for example, a high amount of DNA, in the right side, low amount, and one can use this for quantification. This is always a 1 to 10 dilution. And here one can see easily, the signal is very clear. Let's say it is easy peasy. If this would be pork, everything would be clear, pork would be present. The problem is, for low amount of DNA seen here. Let's say what happens if there's a trace of pork inside, and that's the issue for halal. People are arguing it must be 0. And discussions start, what is 0? We have the CT or CP or CQ values. For example, here approximately 34 for the last curve. What happens at the end? Is it a value, yes or not? Then one should consider these tests are very useful, but they are not halal tests. Some companies claim just that these pork tests are halal tests, but halal, as you know, is more. It is some regional religious aspects which has to be used. And the absence of pork species is only one part of this. So these assays are, let's say, not less, but not more than very useful tools, detection tools, but of course cannot say that this food is halal. The assays are highly specific and meanwhile very sensitive, up to less than 1 ppm in muscle meat. They are usually validated for food, but you can remember for fat it's already difficult. and for other parameter or matrices like leather, feathers, and so on. These assays are not really validated and will be difficult because DNA must be present, otherwise DNA cannot be detected. So just to show what we are talking about, let's say 100% would mean a pure piece of pork meat or meat, which means 1 million ppm. So of course, no need just to test this. This is very obvious. A test for 10% would not make sense because 10%, it's also very clear, one can see it likely. 1%, like for example with currency, let's say $1.01. This is already a quite sensitive detection, and that's the range where one discuss the difference between food fraud or adulteration, if it's more than 1%, or severe incompetence versus contamination. For example, in Europe, we have the standard of 0.9% as a legal threshold, which is a threshold below 1%. But for halal issue, one would like to have more sensitive tests, let's say 0. So just make one more dilution, 100 ppm, and one more dilution. Let's say we are at 10 ppm, and one can go down up to 1 ppm or less. So if talking about results, please keep in mind, these are very sensitive measurements, but it's not 0. It is a technical measurement. So, and then when talking about detection systems in such an auditorium about food regulatory, one should mention, of course, the standards. Here's a standard, is an ISO regulation 28113. I'm just member of this Dean working group for molecular species analytics, and we published 2 years ago one of these ISO standard, which is quite helpful. It has some normative, mandatory aspects and some informative parts, like usually in ISO. And one should keep in mind, I will not give a whole introduction about this. There are a few parts. For halal, one is discussing sensitivity. Should be 0, will not be exactly 0. Sensitivity is defined via the limit of detection, which means all target DNA or molecules should be detected, all of them. Usually one is doing a dilution series with a confidence interval of 95. So it means all 95% will be detected. But what is much more relevant is the dilution in the relative relevance matrix. Let's say pork meat in beef meat, then making homogenization, blending, dilution, and DNA preparation. There's also the limit of quantification possible, LOQ, which is not important here because we are looking for qualitative assays. One should not forget the specificity, which can be done in silico or in practice, so with exclusivity studies, so using different meat samples and being sure that's only pork which will be detected, not to detect some false positives. And also very important, which will be more important, is the robustness. This can be done, for example, via orthogonal variation, just to be sure that assay here in the laboratory in Dubai will show the same result as an assay, the same assay, for example, in Egypt or in Morocco. This can be done with a robustness study here, for example, with a technical specification here on top, which is not standard. It's just a technical specification, but very helpful. So with very weak signals of DNA, one can just do a variation with a PCR equipment, with master mix, lot A, lot B, then doing the normal primer concentration, and primer concentration only 70%, so 30% less. The probe concentration can be less. The volume of the master mix can make plus minus 1 microliter, and the temperature of the thermocycler can be changed plus minus 1 microliter, because the digit will show on a display anything, but in reality there can be some changes. And if everything is really similar, then one can argue this is a robust assay. In addition, one should do an interlaboratory study. We talked about this in the morning. Let's say different laboratories are using the same assay or proficiency tests. I will tell some of them in the next minutes. Being here and having the experts from GSO here available, I should mention also a Gulf standard for immunological methods. I will not talk about this one, but will mention also a second one which applied in July this year. It's a detection of pork in DNA test method, food and food products from GSO, this is 2649, out of GCC. And giving a short introduction in this, there are, is adapted from a Malaysian protocol from 2017. So this describes the CTAP, phenol extraction, how to create all this buffer. We can buy this, this standardization for approximately $30 US. It describes how to use a classical PCR with post-PCR gene electrophoresis. And they are using a 3-step PCR, having results with a limit of detection of approximately 0.1%. And the results should appear in the range of 32 to 34 CTs. Another example, this is not according to the GSO. Let's say that's a commercial test where one can do this DNA preparation now with spin filters, not with wet buffers, with a phenol CTAP extraction. To get purified DNA, one can start 50 to 100 milligram, getting 50 to 100 microliter, and using then the purified DNA, one can measure the DNA, for example, in these cuvettes or nanodrop. We don't recommend it, even though it is mentioned in the GSO standard. One can do this just to be sure that one have enough DNA, and can do a electrophoresis. As you can see, the gene electrophoresis, which can be used also for post-PCR detection, is a little bit laborious and has the risk of cross-contamination. Keep in mind, we are looking for a very sensitive detection, and then cross-contamination can be an issue. Therefore, we recommend to use a real-time PCR, and that's, for example, a protocol using 45 cycles with a 2-step PCR system, having this on farms, this is tropha, the pork, and in hex, which has all the thermocyclers are able to measure an internal amplification control, which will ensure that a negative result for farm for pork is not due to inhibition. So it is a real negative, as good as one can say. So with this assay, I will show the results. That's now kind of main slide. So on the left side, you can see a dilution series of pork meat and beef meat, and then a serial dilution and DNA preparation. It is possible to detect up and below 0.001% pork in other meat. And this is extremely sensitive. So it's really about detecting traces. It's not zero, but very close to this. And on the right side, you can see there was no inhibition. So such an assay is very sensitive, but of course, the more sensitive an assay is, the more risks are. So one has to be careful not to change the settings, because not to detect any artifacts, being aware about contamination. And I noticed mostly in the centrifuge it might happen. One should use it only for food. And of course, finally, one should be careful with its data, even if it's clear. One should not get out, let's say, just sending out of the laboratory a WhatsApp into the world. Oh, I found something. This restaurant is not halal, it's haram, or this brand is not doing well. So because one should have a look, let's say, for the staff about whether the procedure was good, whether enough controls has been done, just contamination could be avoided. The lab management should control this, doing eventually replicates from new samples, getting information where the samples are coming from, how the sampling was done on site, whether there maybe a contamination occurred. And then if it's possible, a positive, someone else should decide what it is it a real positive, how to handle, and of course, how to communicate. This is a normal procedure, but since it's in a religious aspect and there could be more emotion, one has to be careful with this. So one can use also, if the test has been validated, proficiency tests. We talked about this also in the morning. For example, FAPAS is offering proficiency tests for pork, dried material, and DRRR for gelatine, sweets, and other matrices, for example, specific for gelatine and sweets for pork assays. I will show you some data from the very first proficiency round 5 years ago. And you can see there has been 3 samples. So one was positive on the left column, which is green, and 2 has been were without DNA for pork, but gelatin. And you can see also already here, I made there something gray because this has been the names of some commercial companies or methods of some laboratories. Some of them here failed. For example, in the beginning, they had once probably a mismatch in the laboratory. The second one detected only 1 out of 2 samples, so it was not really sensitive enough. Then some samples detected the negative correctly as a negative, but could not detect the positive material. So the test could be quite good for food adulteration, but was not sensitive enough for this purpose. And finally, some assays here are sensitive to detect porcine DNA in gelatin. There will be also a new test round by end of this year, which means in December. And of course, there are other proficiency test suppliers, for example, FAPAS, BPA, LGC, public laboratories. And it makes sense really to validate any kind of method, if it's a standard or just a single method. And same with the proficiency test, what we discussed, one can use different methods. So, for example, also mass spectroscopy, which has been also used in some of the proficiency tests, or immunological assays. And here I will show you, let's say, one result. This was some result from a customer who asked me, oh, we have a problem with such a proficiency test. And here you can see these both curves, clear curves of the positive control. And from one sample, only one was positive, not the other one is duplicate. So probably the laboratory had some tiny issues, and the result was, of course, in this case, presumptive or presumptive in French, and it has to be confirmed. So if it is then positive, then one can argue, yes, porcine DNA was detectable. If this will be negative, one cannot say negative per se, but one can say not detectable, and in brackets, limit of detection, let's say 1 ppm. And with this, I would like to thank you for your attention, just with a picture of some Arabic grill with some nice meat.